everyone. Welcome to today's live broadcast, Direct Microchip CEMS Peptide Mapping Workflow for the Analysis of mAb CQAs. I'm Megan Manzano, the Senior Editor of LCGC, and I'll be your moderator for today's event. We are pleased to bring you this webcast presented by LCGC and sponsored by 908 Devices. I would like to share a statement from our sponsor. 908 Devices builds purpose-built, handheld, and desktop mass spectrometry devices that provide point-of-need analysis for critical-to-life applications. Leveraging their proprietary platform technology, their devices are significantly smaller and more accessible than conventional laboratory instruments. We have a few important announcements before we begin. This webcast is designed to be interactive, and we encourage you to ask questions during the event. You can submit questions by typing them in the Q&A box, which can be found at the bottom of the video player. You can enlarge the slide window by clicking on the small icon in the bottom right corner of the media player. The slides will advance automatically during the event. If you have any technical problems viewing or hearing this presentation, please click on the question mark help widget in the top right of your presentation window. I would now like to introduce today's speakers, Dr. Sara Carillo, Dr. Adi Kulkarni, Dr. Jonathan Bones, and Dr. Erin Redman. Dr. Sara Carillo is the Bioanalytical Research Lead in the Characterization and Comparability Laboratory in NIBRT, Ireland. Sara completed her PhD in chemical science at the University of Naples in 2013, working on bacterial glycoconjugate structural characterization. In 2015, Sara joined Dr. Jonathan Bones' research group in NIBRT, working on CHO cell glycome and the development of analytical workflows for biotherapeutic characterization. Adi Kulkarni is a principal scientist in the strategic marketing team at 908 Devices. Adi has extensive experience in applying CEMS in biopharma characterization workflows. He is primarily responsible for developing novel ZipChip applications and managing strategic collaborations. Prior to joining 908, Adi was an application scientist at Bruker Daltonics. He received his PhD at the University of Massachusetts Boston and was a postdoctoral fellow at University of Southern California's Locher Hydrocarbon Research Institute. He has authored 18 peer-reviewed journals. Jonathan is the principal investigator of the Characterization and Comparability Laboratory at NIBRT. An analytical chemist by training, Jonathan's research group specializes in the development and application of analytical solutions for problems associated with the manufacture and characterization of biopharmaceuticals. This includes recombinant proteins, monoclonal antibodies, and advanced therapies like AAV-based gene therapy, cell therapy, and mRNA, using their core excellence in analytical separations coupled to mass spectrometry. Erin is a principal scientist in the Research and Development Division of 908 Devices. She joined the company in 2016 and has worked to develop protein characterization assays using the ZipChip technology. Prior to her career on 908 Devices, she received her PhD under the direction of Professor J. Michael Ramsey at the University of North Carolina at Chapel Hill, where she developed microfluidic technology for interfacing high-resolution separations with electrospray mass spectrometry. Thank you for joining us today. Before we get started, the presenters would like the audience to answer a polling question. What type of proteins do you perform peptide mapping on? We'll give everyone a few seconds to check in their answer. Okay, getting some feedback. So far, we have monoclonal antibodies as the lead answer. Okay, getting some more. All right, it seems that our primary, oh, here we go. Some more is coming in. All right, monoclonal antibodies is still the top choice, with the second being other. Okay, so with that, I would now like to direct everyone's attention to our first presenter, Dr. Adi Kulkarni. Please get us started.
Thank you for the introduction. I am Adi Kulkarni, a principal scientist here at 908 Devices, and on behalf of the speakers and the panelists, I would like to welcome you to today's webinar. The topic of today's presentation is Direct Microchip CEMS Peptide Mapping Workflow for the Analysis of mAb Critical Quality Attributes. This is a two-part webinar. In the first part, I will provide an overview of the ZipChip technology, and in the second part, Sara will show some great examples of how the ZipChip technology can be used for analysis of mAb critical quality attributes. So let's get started with an overview of the ZipChip technology. What is ZipChip? ZipChip is a capillary electrophoresis-based front-end separation device for mass spectrometry. It is a lightweight nanoflow device that automatically syncs and aligns with the mass spec inlet. The ZipChip is currently compatible with several different models of mass specs from Thermo Fisher, Bruker, and SciEx. The second aspect of the ZipChip hardware is this microfluidic device that is shown in the center of the screen and is also called the ZipChip. This chip is used to perform capillary electrophoresis, and a single chip can perform up to 125 injections. There are two types of chips available, and I will share the details of these chips on the next slides. Finally, we have the ready-to-use reagent kits that cover a wide range of applications. The kits eliminate the need for method development and provide ready-to-use solutions for users. This saves the users a significant amount of method development time, which is otherwise required for separations using some of the traditional techniques. Installation of a ZipChip is a pretty straightforward process. In the image on the left, you can see the ZipChip interface itself. It weighs about 8 pounds and can be easily mounted on and off the mass spec. The image on the right shows the user mounting the interface on the mass spec. The installation takes less than a minute, and after installing the interface, a few electrical and fluidic connections are made. And this is followed by priming of the fluidics on the autosampler and on the microfluidic chip. You can start generating data in less than 30 minutes after mounting the interface. As I mentioned on one of the previous slides, at the heart of the ZipChip technology is this microfluidic chip called the ZipChip. It integrates capillary electrophoresis and nano-electrospray ionization on a single device, making coupling with mass spectrometry a seamless process. Broadly speaking, the chips can be divided into two categories: high-resolution chips for applications that require good separation for thorough characterization, and high-speed chips for rapid analysis. The high-resolution or HR chip is generally used for applications such as charge variant analysis or for peptide mapping, and you will see some examples of the latter in Sara's presentation. The high-speed chips are typically used for small molecule analysis or for getting quick protein ID when high separation resolution is not desired. And these chips can of course be mixed and matched with different BGE kits to obtain optimum results. This slide has additional information on the BGE kits. BGE stands for background electrolyte and is analogous to the LC mobile phase. It is the solution that fills the channels on the chip. The BGE is comprised of a conductive mixture of LC-MS grade water that is mixed with an appropriate mass spec grade organic solvent such as acetonitrile, methanol, or isopropanol, depending on the type of the BGE. It also has a charge carrier, which is usually a mass spec friendly acid, base, or an ammonium salt. The approximate pH of each BGE is listed in the table. And as you can see, the kits cover a wide range of pH, enabling analysis of a range of different molecules under a variety of conditions. And with the exception of oligonucleotides, all other analytes must be positively charged at the pH of the background electrolyte to be able to be successfully analyzed on the ZipChip. The BGE kits also contain the necessary sample diluents that are required for sample preparation. To automate the data acquisition on the ZipChip, the setup also comes with an autosampler that is capable of handling samples in standard formats such as a 48 vial rack or a 96 well plate. The autosampler can be cooled to preserve the sample. The autosampler enables the users to load large cohorts of samples at one time, generate a run queue, and walk away. The workflow on the ZipChip is pretty straightforward as well. Depending on the application of interest, the users must pick the right kit, and depending on the goals of the experiment, they need to pick the right chip, either high speed or high resolution, and load the samples directly on the chip. In most cases, there is no sample preparation involved, and it is pretty much a dilute and shoot approach. The advantage of the ZipChip is that it is highly tolerant towards a wide range of sample matrices, such as high salt concentration and even detergents in protein formulations. And because this is a microfluidic technology, the results can be obtained within just a few minutes. Now let's take a look at how the ZipChip works. The schematic of the chip is shown in the center of the screen. The chip has 4 wells, a serpentine separation channel, and a sprayer corner. And the chip performs 3 main functions. The first important function is sample handling. The autosampler delivers the sample in the sample well. The sample is then pressure injected into the injection cross before turning the power supplies on. Once the high voltages are turned on, positively charged analytes migrate through the separation channel towards the sprayer corner. And this is the second important function of the chip: electrophoretic separations. The third function is nano electrospray ionization. As the analytes make their way to the sprayer corner, due to the high voltage applied at the corner, the analytes are electrosprayed directly into the mass spec. It is important to remember that this is a liquid-only separation, and the channels do not contain any stationary phase, unlike an LC column. The advantage of this is that it significantly reduces the possibility of an injection-to-injection carryover. This data slide shows an example separation of an amino acid standard consisting of 20 amino acids. The system produces very sharp and Gaussian-looking peaks, and most of the analytes are baseline resolved and migrate very rapidly in just under 2 minutes. The separation is based on charge and hydrodynamic radii of the analytes. Lysine, for example, being a dibasic amino acid, has a net positive charge of plus 1.5 at the pH of the background electrolyte and migrates in less than a minute. Aspartic acid, on the other hand, has a net charge of only plus 0.3 at the same pH and takes longer to migrate. So this is how the charge-based separation works on the ZipChip. Now, isoleucine and leucine that have the exact same net charge and also the molecular weight show baseline resolved peaks because of the fact that these structural isomers have different hydrodynamic radii, leading to different migration times. So this is how the charge-based separation mechanism works on the ZipChip. So just as a reminder, the the ZipChip separations are based on charge and shape of the molecules. Because ZipChip uses microfluidic technology, the analyses are performed in a fraction of the time when compared to traditional methods. The same example of the mixture of 20 amino acids that you saw on the previous slide was analyzed on the ZipChip and then compared with a traditional capillary-based CE system. The traditional CE method, as you can see, takes more than 10 times longer for the analysis. The same is true for liquid chromatography separations as well. The ZipChip produces results much faster than conventional LC methods. So the ZipChip is not only orthogonal to liquid chromatography separations, but also produces faster results. ZipChip is truly a versatile platform for analysis of a wide range of molecules. The application space ranges from small molecules to peptides to intact proteins and beyond. The platform has been widely adopted in the biopharmaceutical industry and leading research labs for a variety of different applications. In the last few years, there have been several peer-reviewed publications highlighting the unique aspects of ZipChip for several of these applications, such as peptide mapping, mAb charge variants, or analysis of AAV capsid proteins. Some of these publications are highlighted here. And with that, I would like to thank you for your attention.
Thank you so much, Adi, for such a great presentation. Before we get into our next presenter, I would love to ask a second poll question to our audience. So this question is, if LC-MS is used in your current peptide mapping workflow, what is the scale of your LC flow rate? We'll give everyone a few seconds to answer. Okay, great. We're getting some responses. So far, our most voted answer is analytical flow, followed by microflow. Okay, microflow has now taken the lead. That's great. Thank you so much, everyone, for being so engaged with the polls today. All right, so with that, we will move to our next presenter, Dr. Sara Carillo. Please get us started.
Good afternoon, everyone. As already said, the title of my presentation today will be Direct Microchip CEMS Peptide Mapping Workflow for the Analysis of mAb CQAs. My research group is located in NIBRT, which is the National Institute for Bioprocessing Research and Training, located in Dublin, Ireland. NIBRT is a world-class facility dedicated to training and research needs of the biopharmaceutical sector. And in particular, from the research perspective, we aim to work on research with high impact, addressing and being up to speed with the challenges that are posed by the rapidly evolving of biopharmaceuticals productions technologies. We know biopharmaceuticals can be extremely complex. For a standard scaffold such as a monoclonal antibody, there are several product quality attributes to be monitored through production. That means, of course, from drug design down to drug approval and consequent release of each individual batch. Among the main features that are normally monitored, we find modifications that are specific for the CDR region, modification on the N and C termini, for example, variants in the sequence of the amino acids on the protein backbone, and many side chain mutations, with the most common being the amidation, oxidation, glycation, and surely the N-glycosylation. In the case of more complex biopharmaceutical formats, such as antibody-drug conjugates or bispecifics, the intrinsic complexity can only increase. Monitoring product quality attributes, of course, it's not only an analytical exercise, but it's a critical step in all the phases of the bioproduction. The molecule features can indeed influence the biological activities in vivo and can have a dramatic effect on the stability and the lifetime of the product. Of course, understanding these features can also help to understand and maximize the production and the purification process during the bioproduction. And of course, to monitor these features is a requirement from a regulatory perspective to ensure product quality and safety. Lately, all these leads to more efficient and safe drugs for the patients. With the exponential growth in number and complexity of biopharmaceutical products, it is clear that analytical technologies are required to provide more and more accurate information. Often in a high throughput fashion, to go with the fast pace of bioproduction and provide, where possible, detailed information during the process, with online or at-line monitoring where possible. Biopharmaceuticals are usually analyzed following two main approaches that are either based on bottom-up analysis or on the characterization of the protein at intact level. In most of the cases, the two techniques provide complementary and orthogonal informations that are able to fully characterize the drug product. One of the platforms that we use in our laboratories is made, as you can see here in the picture, so from the thermoscientific Orbitrap Exploris 240, which is hyphenated with the ZipChip TI source, which, of course, in combination with the right VG kit and chip, provide a really flexible instrument for both intact and bottom-up analysis of biopharmaceutical. In particular, in this webinar, I would like to focus on a recent optimization we provided for the peptide mapping workflow to add direct CMS analysis of several biopharmaceutical modalities directly analyzed with the ZipChip unit. Generally, the sample preparation ahead of peptide mapping analysis consists of the relaxation of the protein tertiary structure to enhance the reduction and alkylation of the disulfide bonds afterward. This step is followed by the enzymatic digestion, which is usually performed with trypsin, and then the analysis is performed through LCMS techniques. The LC separation of the tryptic mixture is usually preferred to other separation techniques, as the sample preparation introduces salts that are not compatible with the CE separation, while they are easily separated out using reverse phase chromatography. Moreover, additional cleanup steps may result in low salt sample recovery, as well as higher levels of sample prep-induced modifications, such as, for example, the oxidation. However, CE separation using the ZipChip interface brings some unique advantages, especially in the short analysis time when compared to long LC gradients, the minimal method optimization that is required before running an experiment, and increase the sensitivity. For this reason, we decided to develop a direct method for peptide mapping sample preparation that would guarantee complete digestion and minimize sample prep-induced PTMs. The sample preparation we normally perform for LCMS analysis is summarized in this slide, but the digestion in tris buffer would require another cleanup step for the peptides before CEMS analysis. For this reason, we tried to replace tris with a more compatible buffer, such as ammonium acetate. An additional step after digestion is the dilution of the sample with acetonitrile, reported here in the very last step, which ensures the sample is transferred to the CE interface with the right composition and concentration of 0.5 milligram per ml. In terms of timing, we perform this workflow, the timing that are reported on the slide for each step, which brings the total preparation to 1 hour and 40 minutes. That, with the additional 15 minutes of analysis time, brings the total analysis time to less than 2 hours. For CE separation, we used an HR chip and the peptide VG kit. included the peptide BG included in the peptide kit, of course. CE method is controlled by the ZipChip software, while the DMS is set up from Chromeleon CDS. As I was mentioning before, sample consumption in this type of analysis is minimal, and we optimize the injection volume to be 8 nanoliters or 4 nanograms of material to generate pretty intense TIC that ensures full sequence coverage and good detection of post-translational modifications. Before going into the details of the CEMS analysis, I would like to quickly mention that the workflow was compared with our standard approach by CMS analysis. And to compare the two results, we performed a non-targeted MS comparison, which detected 0 new components and comparable PTMs levels, thus resulting in a very good alternative to standard digestion in TRIS buffer. Now, going back to the CEMS analysis, we started analyzing some standard mAb in triplicate, and the data processing was performed through Biopharma Finder software. We obtained almost a full sequence coverage and reproducible results for the relative abundance of some of the most critical modifications, as you can see in the table on the right. But I also wanted to show you some example of the data quality that we were able to obtain. For example, the modifications that we could monitor include some of the trickiest one, like the penny-K peptide, where the deamidation was completely resolved, as you can see here in the zoom, from its unmodified form. As we can see in the structured ion chromatogram, this is, of course, for the peptide that bears 3 charges. A similar example is visible for the M255 oxidation, which with the LC analysis sometimes can cause on-columns artifacts such as oxidation that occurs during the separation. That usually results in very broad peaks that are affecting the correct quantitation of this modification, while through the CEMS analysis we obtain very sharp peaks that are easily quantified. So starting from the good results we obtained with the mAb, we also decided to test other drugs with different modalities. So, for example, we tested either like IgG2, IgG4, fusion proteins, and of course, other types of IgG1. The sample preparation and the CEMS method that we applied were exactly the same in all the cases. And in all cases, we also obtained good results and sequence coverage. I just wanted to show you some example of the results that we obtained. One of the molecules tested was the fusion protein at Livercept, which is known to possess 5 different N-glycosylation sites for each of the chains. Some of them have very elevated acylation levels. In the slide, I'm just reporting a summary of 2 of them as a graph bar of their relative abundance. And the levels obtained were very similar to those present in the literature, highlighting high level of galactosylation, for example, for the FC and glycan, and very low level of fucosylation for the sialylated form of the second one, with the most abundant species being the S1G1 species. A second example that I want to give you is the results that we obtain on nivolumab, which is an IgG4 monoclonal antibody. In general, we use, of course, for all the data analysis, a 90% confidence score and 5 ppm of mass accuracy tolerance for any data analysis. This is really to be confident, of course, in our results. An example of the quality of the mass spec data obtained is shown in this slide. Of course, one of the critical quality attributes that we monitor for nivolumab is the formation of the pyroglow on the N-termini. The modification was correctly detected and quantified, and it's possible to appreciate the peptide coverage here in the middle of the slide, as well as the comparison of the predicted and the experimental MS2 spectra, which are really alike. And so we can surely say that this peptide was detected with great confidence and correctly quantified. In the last example, we focused again on an IgG1, such as rituximab. This monoclonal antibody has a deamidation hotspot, which is critical for its activity, being located in the asparagine 55, which is located in the CDR region. Although its abundance is below 1%, we obtained confident identification and quantitation. Also thanks to the quality of the data shown in the zoom of the two spectra on the right. Of course, in this case, we can see the isotopic distribution here in the middle of the double charge unmodified and modified peptides. So, in conclusion, I hope I presented you a few examples of a simplified workflow that avoids additional cleanup steps for direct analysis of peptide mapping through CEMS. And the whole workflow was completed in less than 2 hours using minimal amount of sample, as low as 4 nanograms per replicate. And we also proved that the workflow, of course, can be applied to multiple biopharmaceutical formats and IgG subtypes, but can, of course, be optimized further where, for example, longer digestion times are required. To conclude, the data processing demonstrated that CEMS analysis performed in this way allowed the confident measurement of the most common critical quality attributes, thus demonstrating the suitability of high-throughput peptide mapping analysis performed through the ZipChip interface. And with this, I want to conclude and thank my team at NIBRT, Silvia Milan-Martin, Rachel Ronan, which initiated this project, and of course, Dr. Jonathan Bones, which is our principal investigator. And also our collaborators at 908 Device, KU, Erin, and Adi. And really thank you for your attention.
Thank you so much, Sarah, for such a great presentation. Before we head into our Q&A session with our other panelists, we have 2 more poll questions for the audience. So the first one we have here is, for your current peptide mapping workflow, what are the major challenges or bottlenecks? Please check all that apply. We will give everybody a moment to answer. Okay, just taking, here we go. Took a little second to get the results. Okay, so far we have method development and sample availability as being the most challenging or bottleneck. Okay, data processing and interpretation has jumped. Okay, we seem to have a range of problems from everybody here. But data processing and interpretation does take the lead. Great. All right, so we'll move to our last poll question of this session, which is, how many peptide mapping samples are you currently running per week? We'll give everyone another minute to answer. Okay, great. Okay, so a large lead right now is 0 to 20 peptide mapping samples per week. Give everyone another minute. Okay. All right, the lead is still held at 0 to 20 samples per week. Thank you so much, everybody, for participating in the polls. And now we will get ready for our Q&A session. So with that, I would like to welcome Adi, Sarah, Jonathan, and Erin for our panel discussion, where they will be taking questions from the audience. Before we get started on the question and answer session, I would like to remind our audience how to submit questions. You can submit questions by typing them in the Q&A box, which can be found directly below the video player. So our first question for everybody here is, what is the maximum length the peptide can be for analysis? Is 48A too long?
I would say Sarah, do you want to take that?
Yeah, I can do that, Erin. No, it's not too long. I think in our sequence coverage map, we were showing even longer peptides, up to kind of 57 amino acids. It all depends also on the charge that they bear, so that will affect their mobility. No, surely 40 is not a limit.
And I would remember that the separation is free in solution. There's no stationary phase. So provided it has enough charge, it's going to go through the chip, no problem. We use similar separation conditions to do intact proteins, and they make it through perfectly fine.
Wonderful. Thank you both, Sarah and Erin, for taking this one. The next question we have here is, can the ZipChip be used for oligos?
Yeah, I can take that one. So the short answer is yes, the ZipChip can be used for analysis of oligos. Our standard chips used for analysis of peptides and proteins, they are polymer coated, right? For oligos, we use a different chip chemistry. So we use the bare glass chips for analysis of oligos because bare silica is negatively charged naturally. So that helps in repulsion of the negatively charged oligos and prevents any adsorption on the walls of the channel. The ZipChip methods are fast, and more importantly, they do not use any ion pairing reagents. And you can get very sensitive positive mode ESI data within just 5 or 6 minutes on these oligonucleotides. And our customers really like this method because there's no downtime associated with cleaning all the ion pairing reagents from the mass spec and LC before you can switch your application. So there's no dedicated system required there.
Wonderful, thank you. We have another question here that sort of ties into the one, some of your answer, Adi. What is/are the chip materials? Did you observe any absorption on channel walls?
I can take that one. The chips are made of glass, and like Adi described, we lay down a coating on the chips that we use for peptides and proteins, and that prevents a lot of absorption that you would potentially run into with introducing peptides or proteins to glass. Also, we're running in high organic conditions, and this sample is also in high organic conditions, and that helps prevent a lot of sticking that you may encounter with something that's mostly aqueous solution.
Wonderful, thank you, Erin. We have another question here about oligo, so I'll ask that to the group. With oligo analysis, are there any modifiers to minimize salt adducts in the ZipChip?
So we do analysis in positive mode ESI. All of the reagents that go in our oligonucleotide background electrolyte, they're all mass spec friendly reagents. We do not use any ion pairing reagents at all. We rely on just utilizing some of the cleanest mass spec friendly solvents and reagents out there to successfully reduce the adduct levels. And our own work has shown that the levels of sodium and potassium can be below 10% relative to the protonated adducts if the right reagents are used.
Wonderful, thank you. The next question we have here is, can we use ZipChip with any mass spectrometer?
So the ZipChip is currently compatible with mass spectrometers from Thermo Fisher, Bruker, and SciEx. We are limited to these 3 vendors for now. That might change in the future, but currently we are compatible with SciEx, Bruker, and Thermo mass specs.
Great, thank you. Our next question is, 30 minutes seems short for digestion. Is this suitable for other modalities?
I can take this one. Yeah, so for sure, there might be some molecules for which the digestion protocol needs to be optimized. But in general, we saw like a good flexibility of this workflow for different modalities, reaching always good sequence coverage and perfect digestion. So I would say it's a good digestion time for most of the modalities that are out there at the minute.
Great, thank you. We have another question about oligos here that says, how big of a synthetic oligo can be analyzed?
Yeah, so like I mentioned, we do not use any ion-pairing reagents. So if the size of the oligos goes up, its propensity to form adducts, alkaline metal ion adducts, also increases with size. If you want to look at the M plus H ions, we've successfully analyzed oligos that can be as high as 70 or 80 mers, while getting sodium levels up to about 40%, assuming all the clean reagents have been used. We haven't really tried any oligos that are bigger than that.
Great, thank you. The next question we have here is, I feel the bottleneck for this fast method is time resolution for MS, considering maintaining of mass resolution. Is it possible to obtain sufficient MS and MS spectrum in each analysis to clarify each quality attribute?
I can do this one, Megan. Yeah, so for sure, like the analysis that we have performed that were filtered to only consider the species that contained good quality MS data. So our confidence score was up to 90%. So for sure, we cannot use exactly the same MS method that we would use for CMS analysis, but there are some parameters that we can tweak to obtain a good amount of MS events, even with this fast analysis.
Wonderful. Thank you, Sara. Our next question is, have you developed ZipChip compatible workflow for other enzymes besides trypsin?
I can do this as well. Not at the moment. We are working on them because we know that there is need for other modalities of digestion. So for sure, it's something that we are working on, but no at the moment.
Follow up, and I would speculate there's no particular reason why another enzyme wouldn't be compatible. Maybe it would require a few minor tweaks, but I would expect it to be fine. I know we've done some pepsin digestion in the past. So I would expect that to be maybe not trivial, but not impossible as a different enzyme.
Fantastic. Thank you. Our next question is, do you see any digestion artifacts from performing the digestion in ammonium acetate versus TRIS buffer?
I can take this one. No, as I showed in the slides, we see a very good comparability between the tris and the ammonium acetate digestion. So there was nothing really coming up because of the different buffer used for the digestion. The digests were pretty identical. So no, I would say it's not really influencing the digestion efficiency and the results.
Wonderful, thank you. Our next question here is, can the same sample be run by LCMS?
Yes. There's the small caveat is that our samples are in high organic by the time you load them into an autosample vial. So you could, modify the procedure to not put them in high organic or dilute them so that they're at the appropriate low organic that you would want for chromatography.
Okay, fantastic. The other question that we have here is more of a question for me, which basically says, how can I get a copy of the presentation today? So for everyone listening, if you have signed up for this presentation, you will receive an email sometime after this presentation has ended with a link to an on-demand version of the presentation. So you will have a copy of it. Okay, so with that, we would like to wrap up today's presentation. I want to thank the audience so much for participating in today's event, especially with the poll questions, and for asking our panelists some wonderful questions. I would also like to thank our sponsor, 908 Devices, for making today's educational webcast possible. We would like to thank everyone in the audience to participate in a brief survey. The survey will appear on your screen after today's presentation has ended. You will receive an email alerting you, and this webcast will be available for replay. We invite you to forward that announcement to your colleagues who may have missed today's live event. Thank you to everyone for joining, and we'll see you next time. Have a great rest of your day.